So I am presenting immune and inflammatory proteins in cord blood as predictive biomarkers of retinopathy of prematurity in preterm infants to audience B. So essentially what happens when babies are born prematurely, the retina, which is the kind of tissue in the back of the eye that allows us to see, it's almost akin to the film on a camera, is not fully formed. The areas that don't get enough oxygen, they actually grow these kind of abnormal blood vessels which sounds like a good idea, we want blood vessels to kind of help supply oxygen, but what actually happens is these ones are really crappy blood vessels. They kind of cause a bunch of pulling on the retina and can actually lead to a detachment. This is an issue because these infants can develop really big vision problems for the rest of their life. So essentially what they were trying to do in this paper that I'm explaining was when babies are born, you can actually sample some of the blood from the umbilical cord. This is important for a number of things in the field of obstetrics, but for ophthalmology, we can actually measure some proteins in the blood that are involved in some inflammatory and immunologic pathways, essentially how the body fights disease, and see if these correlate at all with the progression of this disease called retinopathy of prematurity, essentially being how your retina is affected when you're born too young. So in this study, they looked at 110 of these infants who were born less than 32 weeks, which is actually that 32-week mark is established by the pediatric ophthalmologist as kind of a good benchmark for maybe we should look into the back of these babies' eyes and see if they have any signs of this ROP disease. And they looked at a number of these markers, like I said, but what they found is that this one protein called IL-6, which is involved in kind of cell signaling at a very kind of deep immune level, was associated with the severest progression of retinal disease of the infants. This other protein called C5a was also associated with a particularly severe type of ROP that actually required laser treatment to kind of spot weld the retina back into place. Essentially, the conclusion of this paper is that IL-6 and C5a, when taken from the blood of the baby's umbilical cord when they're born, might allow for monitoring of this disease and possibly which infants are going to need this laser treatment to kind of basically push the retina back into place, seal it down, and allow it to sort of reform. Unfortunately, this disease is kind of a bad prognosis, there's a lot of risks associated throughout the body when a baby is born early. Usually their lungs don't work as well. They can have a lot of issues with seeing as well, like with this disease. They might kind of have issues with their gut. Their gut can get infected, but this is kind of the eye problem that we watch out for.